Thank you for watching this presentation. We have nothing to disclose. Nitric oxide, NO, is widely recognized as a representative gas transmitter and an endothelium-derived vasodilation factor. In the lower urinary tract, NO is well known as a relaxation factor in the urethral and prostatic smooth muscle. NO is also produced in the brain and plays multiple roles as a neuromodulator. It is previously reported that centrally administered L-NAME, an inhibitor of NO synthase, reduced intercontraction intervals (ICI) in awake condition as shown in green. On the other hand, in anesthetized condition, L-NAME prolonged ICI as shown in pink. These data indicate that brain NO seems to have both inhibitory and facilitatory roles in regulation of the micturition reflex. However, brain mechanisms for brain NO-mediated regulation of the reflex are still unclear. In this study, therefore, we investigated the effects of centrally administered SIN1 and NO donor on the rat micturition reflex and the mechanisms of the SIN1-induced effects on the reflex, focusing on brain glutamatergic receptors. This slide shows the experimental procedures. In urethral anesthetized male rats, SIN1 was ICV administered 30 minutes after ICV pretreated with carboxypTIO, an NO scavenger, or glutamatergic receptor antagonists, MK81 and DNQX. And we performed continuous CMZ and single CMZ. Then I'll show you our results. First, we investigated the effects of centrally administered SIN1 and NO donor on urodynamic parameters, intercontraction intervals ICI, and maximal voiding pressure MVP. As you can see, this donor dose-dependently reduced ICI without affecting MVP. This table shows results of single CMZ. Compared with the vehicle-treated control group, SIN1 significantly reduced single voided volume and bladder capacity without affecting post-voided residual volume or voiding efficiency. These data suggest that centrally administered SIN1 induced frequent urination. Next, we investigated the effects of central pretreatment with PTIO and NO scavenger on the SIN1-induced ICR reduction. The SIN1-induced ICR reduction was significantly suppressed by PTIO. So, these data indicate that centrally administered SIN1 reduced ICI through NO production. As you know, glutamate is a representative excitatory neurotransmitter. Therefore, we investigate the effects of MK81, an NMDA-type glutamatergic receptor antagonist, and DNQX, an AMPA-type glutamatergic receptor antagonist, on the SIN1-induced ICR reduction. As shown in the upper graphs, pretreatment with MK81 dose-dependently suppressed the SIN1-induced ICR reduction. On the other hand, as shown in the lower graphs, DNQX showed no significant effect on the SIN1-induced response. These results indicate that centrally administered SIN1 reduced ICI through brain NMDA receptors. This slide shows summary of our results. These data indicate that NO centrally induces facilitation of the micturition reflex through brain NMDA type glutamatergic receptors. Thank you for your attention.